So I'm really excited to have the opportunity to tell you actually about the broad field of my research, keeping it very high level today. Do we have the slides? Yep, we're good. So I'll be telling you about imaging, and we're not going to be talking about medical imaging. You've heard bits and pieces throughout the conference, especially Eric Tuppel talking about radiology kinds of medical imaging. Here we're actually going to be talking about cell images. What if a diseased cell could tell us what's wrong with it? What if it's the case that those intracellular structures that are so beautiful to view, if you're a cell biologist at heart like myself, what if those structures could tell us the pathways that are active in the cell, the diseases, pathways that are dysfunctional in a cell? And of course, there's some strong evidence that images generally and facial and structures can generally report on diseases. Let's take an example of a pediatrician looking at children's facial structure. They can often diagnose a disease with a single glance at a human patient's face. And pathologists, same principle. Structural changes indicate something's wrong functionally. And they have to zoom in. They might have to take samples of tissue and so on. But the same principle applies. Now, if you're a researcher, we're a little bit stuck, right? Because we can't be doing experiments on humans, and we can't be necessarily doing very large-scale experiments with tissue samples, just very expensive and time-consuming. So what can we do? The premise of my lab is, what if we could readily take some cells, grow them in a lab, and observe their structure? This led us to create the cell painting assay, which was a collaboration with Stuart Schreiber back in the day, where the idea is let's paint the cells with multiple stains that can label all kinds of things in the cell, all your favorite organelles, and then we can do testing at incredible scale. And if we can test cells at scale, we can take samples, test drugs one by one by one. In a pharma environment, you can easily do this cell painting assay for 1 million compounds, not that expensive. And now, at very large scale, we can potentially identify diseased cells, and we can identify drugs that can turn those cells healthy again. So we'll look at some examples today. First of all, how do we do this? How is this possible? And the answer is we measure everything in the cell, which often involves deep learning. We use software to identify each of the structures within the cell, and then we measure all these properties. How many speckles does it have? What are the shapes of the nucleus, of the cell borders, the sizes, intensities, textures of all these different stains that we've added to the cells. And because this is fully automated, we can run this at scale. We can test hundreds of thousands of samples in multi-well plates, like shown here. And for the software side, you'll see we produce thousands of features for every single cell. It's a tremendously rich kind of data source. To do this, I invented this software cell profiler during my postdoc at MIT, and it has grown up with the field. So we still use it today. It now has deep learning capabilities and is popular throughout pharma and academia. So you might wonder, okay, sounds neat, you can produce profiles, they're like mRNA and protein profiles, but why do that when you can just do omics directly and get more molecular information? And here's a couple of reasons why. First of all, the image-based profiling is 10 to 1,000 times cheaper than other omics technologies, especially at the single cell level. And what's exciting and honestly completely shocking to me is that we get similar predictive success. So my lab now is entirely computationally based, it's machine learning based. And when we attempt to do different tasks with data from cells, we get comparable results using this humble little cell painting assay as compared to mRNA profiles and protein profiles. And excitingly, the data is quite complementary. So there are some results that you could get with either type of profiling, mRNA or protein versus images. And then there's others that are unique to each of the different types. So it's really lovely when we can do experiments that involve multiple of these different profiling types. But only imaging can be done at serious scale, and that lets us do different kinds of experiments. Okay, so I live in this world of automation, but I've been asked to give a little bit of personal context here. And so I grew up with a profound understanding of the value of hard work. I grew up on a farm in Indiana, and I developed a strong ability to make myself do tedious tasks, which I think is very useful as a professor with the paperwork and administration and so on. But when I went off to graduate school, I spent an entire summer moving a microscope stage around, measuring cells one by one. I would actually get seasick, so I could only do it about 4 hours a day before I'd puke. But I got to the point where, you know, it was pretty miserable. It was pretty miserable, but it was still better than farm work. Because it was air conditioned. Our Indiana summers are very much like what you've been experiencing these past few days. So I was pretty happy, actually, on balance, but I was even happier when my professor got a license for some software to automate the microscope stage. So I went to town and very quickly learned how to code and automated the microscope as well as the image analysis so that I could let the system collect in 8 weeks what I had previously taken 4 years to do during my thesis work. I was absolutely hooked. So when I launched my lab at the Broad, my mission was to turn this love of automation and computation towards drug discovery. Can we speed up the individual steps of drug discovery using not just the physical automation of the robotics, but also clever biotechnology techniques and also AI? And can we even surpass human capabilities? And this is the real excitement that we heard this morning from Mark, as well as yesterday from Eric Topol and many others. That's the real excitement here, is can we do more than what we could do if we didn't mind, tedious work so much? So I'll tell you about the advancements that we've made, a few of them in my laboratory, but I want to put this in context as you've been hearing about AI and drug discovery, and we caught a glimpse of this earlier today. AI is getting better at some tasks faster than others. So if you want to predict which amino acid you're going to get given a triplet of mRNA nucleotides, of course, there's a simple rule you can follow. You don't need machine learning for that. If you want to predict how much mRNA will be in a given cell with a given genome under given conditions, or you want to predict what proteins will be present given a certain mRNA, we have a lot of data now, a lot of public data, shared data, and a lot of techniques that make it possible to catch this kind of data. And that allows us to be able to train machine learning algorithms. As I'm sure everyone is aware, the data is really key to being able to build these models that do a good job making these predictions. So some of these tasks have really gotten better fast. And protein structure, we also heard about today, is one of the shining triumphs of really the open shared data that made it possible to train models to predict structure from sequence. Not a solved problem, but definitely we are so much further along now than even 10 years ago. Okay, so that's all great. We can predict a lot of things, but this is where it falls apart. How do we get from there to curing things? And it's messy because there's not simple rules we can follow to try to connect proteins to diseases. There's not enough direct data that we can train on, and we just can't experiment on humans, at least outside of very small constrained clinical trials. So I would like to propose cell phenotypes as a way to bridge this gap between what are genomes and transcriptomes and proteomes doing and how do the disease phenotypes end up. And I think AI can accelerate this. So how can we do this? Here's an early example from the University of Utah. Here they took some cells, they added some stains, knocked down a gene that is known to cause cerebral cavernous malformation. And the difference is super obvious by eye. You don't need fancy image analysis, but once it's automated, you can certainly scale this to a large degree. So whether a phenotype is obvious or subtle, once you find one, you can then screen drugs to see, are there any drugs that make these unhealthy cells look healthy again? And they used this approach to identify drugs that caused the cells to look healthy, and then they scaled everything up within a startup called Recursion. It used my lab's technology of the cell painting assay, as well as the cell profiler software that I had created. So I didn't found the company, but I was involved on the scientific advisory side since the very beginning, and it's been just thrilling to see their progress and the impact on the world. So now they've identified thousands of screenable image phenotypes and hundreds of potential therapeutics, and have launched multiple clinical trials. Now, back in my lab, we've taken this approach for a number of different diseases, but in slightly different ways. So here's an obesity example where, of course, there's not one gene that you knock out and end up with metabolic disorders. So instead, we take cells from patients who have been calculated as having a high or a low risk for developing metabolic disorders. And that's this polygenic risk score is based on genome sequencing. And by taking those patient cells, they adapted cell painting, added a lipid dye, as you can see these big green droplets here, and identified a very obvious difference in the morphology of the cells here. We can now embark on a drug screen to identify potential therapeutics based on taking high-risk patients and turning them low-risk looking. We can also do a genetic perturbation screen to try to find new targets in this space. And this is a collaboration with Melina Klaus-Schnitzer's lab, who leads in this field. Turning to neurobiology, neurological disorders have been really fruitful for morphology phenotypes, and maybe in a place you would not expect, which is fibroblasts, skin cells. So in our own collaboration with Bruce Cohen at McLean Hospital here in the Cambridge area, we found that mitochondria are less dispersed when patients have bipolar disorder. And again, once you see there's a difference, you can test chemicals to find those that look promising. We currently are analyzing data from a screen that the NIH NCATS Center has done for us in order to look for potential novel routes to treating disease areas which are very difficult and very genetically challenging to understand. Now, turning to rare diseases, we took a totally different approach. Here, we tag proteins so that they glow green, and we introduced two versions into cells. On the top, we have, this is where this protein normally is located in cells, and on the bottom, we've introduced a mutation that is known in each case to cause a human disorder. It's very causative, single gene, very simplistic, let's say, rare diseases. And in each case shown here, there's a difference between the healthy and the diseased state. So in the Kopak-like at the right, we see that normally this protein should be smooth, but when this mutation is present, the cells get speckly again. What's really exciting is just how scalable this is. I don't need to know anything about any of these disorders. I don't need to know anything about what these proteins' normal function is. I can just make all of them. And so that's what we're on our way to doing. We started out with 3,500 genetic variants associated with a whole slew of different human diseases. We found 250 disease phenotypes, and now we can scale this up, and we're scaling it up in two ways. The first is by an NIH IGVF consortium where we're testing a much larger set of variants to find more and more disease phenotypes, but that still makes our bottleneck even worse. So doing one drug screen in an academic environment is not particularly easy to get funded. Doing 250 is impossible, and doing the 3,000 to 5,000 that we'll likely get from this larger scale, also pretty impossible. So here we had to turn to a clever biotechnology technique, which I won't describe today, but developed at MIT in Paul Blaney's lab and adapted by JT Neal, is allowing us to pool genetic reagents. So it's pooled optical profiling, which allows us to take hundreds of these diseases, combine them into a single well, test one drug at a time, and see whether it can revert the given phenotype. For that, we're launching a VISTA consortium of pharmaceutical companies who are interested in taking already optimized compounds and repurposing to see, are there disorders where these compounds could, or these existing drugs could be useful for patients who otherwise really have no hope. And then finally, turning to a cancer example, this project led to my first startup company, Sizonk. And here we carried out a novel kind of virtual screening where, just like the other cases, we first have to find a phenotype. So here we did it by suppressing the YAP1 gene in cells. And I don't blame you if you don't really see the difference between these two panels. Trust me, the computer can see it. And if humans stare long enough and are taught properly, you can eventually see it yourself as well. But what's neat is that it's just such a unique, specific phenotype that we are able we could do a drug screen. However, again, since we're in the academic environment, it's hard to get that funded. So we devised a workaround. And basically, it takes, instead of developing a custom assay for each gene that we might, each pathway we might care about, and then doing a physical drug screen that's on the order of a year's worth of work, plus in a drug company, maybe several millions of dollars to do the screen, instead we turn this into a virtual computational screen, which takes an afternoon. And the idea is, have we ever seen this phenotype? Have we ever seen this change in morphology in any of the images in our giant database of chemical perturbations? And if we find a match, we can cherry pick just those few dozen compounds and then test them in whatever assay makes sense. So in the case here, collaborating with Karen Eisinger, a professor at Penn, we identified compounds that can kill sarcoma cells, otherwise really challenging to do. We didn't just stop with a viability assay and cultured cell lines, but went to a very unique tumor microenvironment assay that she has developed, which involves the matrix, which is super important when you're looking at solid tumors. And the positive results from all of that caused us to launch Sizonk, where we are building an AI-driven map to identify new solid tumor targets based on this novel class that Dr. Eisinger had discovered, and also finding the promising compounds that will go along with them. Now, even though I'm most well known for my work helping start recursion, I was really only involved on the science side, so I had nothing to do with the business side. So the MIT Faculty Founder Program was absolutely critical for, I could have figured it out myself, but it was just so much more efficient to go through the program and learn from the experts that were, who had tread that path before. So together with the initial funding from Royalty Pharma sponsoring the prize for this, it was crucial to us opening up our labs. And we've now raised our pre-seed round, and it's just been an opportunity of a lifetime to make a difference for patients with incurable solid tumors. Okay, so I've only shown you a teeny fraction of what you can do with images today, mostly focused on this one use case, finding disease phenotypes, reverting them with drugs. I promise you there's a dozen other things that you can do, but one last vignette I will share has been in the news lately. Predicting tox is just a disaster. We're really bad at it as a society, even in pharma, where we put tons of resources in trying to discern this question. And we're very slow at it, which means hundreds of thousands of compounds that are used in chemical use have actually never been tested for safety. We really don't even know. And so the government has recently announced, you've seen this in the news recently, a move away from animal testing, but the question is, what do we move towards? Thankfully, it's not just last month that this has come up. The US government, as well as Europe and Japan, have been moving in this direction for years. And so what can we do about this? We're creating a consortium called OASIS. It has more than a dozen agrochemical and pharmaceutical companies that are collaborating in order to test multiple readouts, cell painting, transcriptomics, proteomics, in multiple human cell models, including some organoids and liver on a chip, as well as some cheaper cell models, in order to see, does any of that data allow us to predict liver toxicity? If yes, and especially if it's cell painting, a pharma company could readily run this assay on every compound they have in their library, not terribly expensive, and have that information very early in the drug discovery process. So I hope you have enjoyed this jaunt through how AI is being used to interrogate images and discover the causes and cures for disease.